Welcome everybody to Biology B. We're talking about lesson 1-1 of our first unit on gene expression. We're going to be discussing how proteins work to express DNA. Today's target is to focus on the understanding that all body cells in one organism have the exact same DNA, but they have different shapes and functions because the genes are expressed differently in different tissues. So, for example, in class last semester, we learned about skin or muscle or blood cells. Those are different tissues and they have different cells with different structures because they have different functions. The process of getting to that differentiation we learned about when we talked about the DNA and genes unit. And we learned about specialization and differentiation. So cells are specialized in different tissues because even though they have the same DNA, these are the words you need to highlight on your guided notes, even though they have the same DNA and the same genetic code, they do not always produce the same proteins, causing them to have different functions. So remember, it's the proteins that are made from the DNA that determine who and what we are and how we do it. So in this illustration here, if you remember, this is the nucleus, and the DNA inside the nucleus is the genetic blueprint. The DNA is what tells the cell, you should make some proteins. And the cell says to itself, oh, okay, I think I'll make some proteins. It doesn't have an option. It does what it's told to do by the DNA. It reads the code that's there, and that's what's produced. So to hit today's target, we've got 3 success criteria. We need to be able to feel comfortable and explain how proteins are built on the instruction of DNA through transcription and translation. Now, the process that we learned about last semester for this is called protein synthesis. If you're new to me this semester, you might have a different name or number for that unit, but it's the one where we discussed how DNA is converted to mRNA, and then the mRNA is translated into amino acids. If you need a refresher of that, please reach out to me early. The second success criteria is that we need to be able to describe evidence that all body cells contain the exact same genes in the nucleus. So all body cells of one individual are going to have the same genes on their DNA strands. So we're going to talk about the evidence for that. And then the third is to describe how cell specialization is the result of gene expression. By the end of this little video, you should be able to explain, describe, and give evidence for these 3 success criteria. Alrighty, let's step back for a minute and look at the big picture, the big idea illustrated. We know that every cell in an organism has the ability to produce every protein that an organism could need. How do we know that that's possible? Now remember, we know that each an organism's DNA has the same sequence and it contains the same codes for all the same proteins. We learned about that when like I said, we went through protein synthesis, that unit where we learned about how DNA is converted, synthesis, is converted to proteins. So DNA in the nucleus is copied, a single strand called mRNA is produced, and that leaves the nucleus. The mRNA travels to the ribosome, which is another form of RNA, but it's an organelle that's freely floating in the nucleus. The mRNA gets fed through the ribosome, and the ribosome matches certain codes on the mRNA with certain amino acids that form a protein. So this long strand of beads on a chain, for example, Those are amino acids, and those are the building block for proteins, a polypeptide chain. And it's the original DNA code that will determine what the protein will become. Sometimes we look at the roadmap of the big picture and we find out where we are to figure out where we have to go. So here's where we've been. We already know about stem cells. Remember that stem cells come in one place. You can find them from the dad's sperm when it fertilizes the mom's egg, right? And what you get is a zygote, right? A fertilized stem cell. That stem cell will make more cells just exactly like it, but all of those cells are blank in function. They are a clean slate, but they all have the same genetic code because they all have the same DNA that came from the same original stem cell. Now, whether each of these cells become a nerve or a brain cell or an enterocyte and a digestive cell, osteocyte and bone, whether they become cardiac cells to the heart or hepatocytes to the liver, whatever their ultimate function is, they all have the same genetic code. They differentiated from the same zygote and fertilized egg. That's what we see happening up there. This is something we talked about in meiosis, and it was in DNA and genes. So we talked about this with differentiation, growth, and development in our DNA and genes unit. We also referenced it again when we compared mitosis versus mitosis versus meiosis. We can go back and review those if we need to. But not every cell produces the same proteins as another cell, right? You have different tissues in your body, and those tissues, those cells in those tissues don't make the same proteins, and sometimes that's a really good thing. For example, our eyes, they don't make digestive enzymes like HCl, like stomach acid. Your eyes don't make stomach acid. Your heart cells don't make melanin that control the pigment in your skin. And that's okay. But the factors that do control what genes are expressed in a cell are called transcription factors, regulation factors we're going to get into. They're not always used. So there are large patches of the DNA strand that aren't used. Now you can access one of these two recordings from somebody else if you'd like to review more on the subject. Bozeman is a great resource. He tends to get a little bit more in depth than we probably need to go, so just a heads up. But let's compare this chart here that we're looking at. We are looking at 3 types of body cells. This is a red blood cell. A red blood cell is shaped like a shallow dish because it needs to carry oxygen around in your bloodstream to your different tissues. An eye lens cell is shaped like this because ultimately that's what the lens is shaped like, and they sort of stack up on each other to make that shape ultimately for the lens cell. Okay, a pancreatic cell is going to express or excrete some digestive enzymes, right? So they play a role in digestion. So when we're looking at this table, we also need to take into account there are 4 genes we're discussing: the hemoglobin gene, the crystalline gene, the insulin gene, and the rRNA gene. Now, if we remember what a chromosome looks like, right? This one happens to be duplicated. That's why I'm copying it like this. There's a centromere in the middle. Do you remember that there are sometimes there's a gene over here this band, this loci for a gene. There might be a gene located here. There might be a gene located here. And there might be a gene located here on this chromosome. They're not, these 4 genes are not all on the same chromosome, but for my intents and purposes, I'm going to do that. So for example, the hemoglobin gene, all cells have these, right? But you'll notice that the gene that makes the protein hemoglobin that binds oxygen, it needs to be present in red blood cells, but it doesn't need to be present in your eyeballs or in your pancreas. Those 2 things don't travel around your bloodstream, and there would be no need for them to deliver oxygen. So they don't need to put the energy out to make a protein for hemoglobin. Likewise, crystalline, you wouldn't find that structure that makes your lens in your red blood cell or your pancreas, but you do need it in your eyes. While the embryo is making your lens, your eye lens. Insulin is the chemical that breaks down sugar. So as you eat sugar, insulin helps break down the sugar, and it basically is a chemical messenger to tell your body when to stop breaking sugar down. So you don't need that in your red blood cells. Your red blood cells don't digest anything. Your eyes don't digest anything, but you do need it in your small intestines. Your small intestines need to know when to stop aggressively pursuing sugar. Also, the rRNA gene codes for something called RNA polymerase. Now, we learned about this enzyme a few times last semester. Let me fix that. But RNA polymerase is an enzyme because it has an ASE ending, and it builds polymers of RNA. So it's the enzyme that puts together and builds the RNA that makes all this other stuff happen. So you have to be able to use and build and make RNA in all of these tissues. That's why it's expressed in all of your tissues. When we are talking about gene expression or repression, if the terminology we're going to be using is if it's expressed, it's on. So if the gene is expressed, then the protein will be made. If the gene is repressed, the gene is off and there will be no protein. So it just won't even be made. The body wants to be efficient. A cell wants to be efficient, and it's not going to produce and create a substance that it's not going to need. It's the way it helps conserve its resources. When we compare specific tissues, muscle, for example, versus skin cells, muscle cells, if you think about the way a muscle looks, if I even drew it like this, and we picture that this is a bicep, that's a guy clenching his arm, right? Here's the other arm, here's his head. If you curl your arm up and your bicep gets big like that, that's kind of how these muscles are shaped. Put a bunch together, and that's what a muscle looks like, right? And so they have a similar structure to their function. Their function is that when they get shorter, when you contract a muscle, it gets thicker through this region. That's why they're shaped that way. Skin cells are stacked on each other, much like tiles on a roof. And they do that to block, they act as a barrier for the outside. And because if you get an abrasion and you lose a couple layers, you still have these skin cells to act to prevent invaders from coming in. If you think about how often you lose skin cells, you want to have a bunch of layers that are kind of the first line of defense. So these 2 types of tissue, muscle and skin, they look different and they act differently because genes are expressed differently, even though, and that's the key here, even though they have the same DNA molecules, they're expressed differently. Okay, a couple of things that you need to know from your guided notes here. Again, when we're talking about growth and development, different genes are active during different stages of development. You, for example, are probably going through, well, even we think about the zygote, when the egg is first fertilized and it's forming this um, collection of specialized cells that will eventually form a head and an arm. And there's two legs and there's the spine of the zygote of the infant in utero. Different genes are active during that part. But there's also genes that are only expressed at certain times. That's controlled timing. Like, for example, you're going through a stage in your life right now where things are changing a lot. You're growing hair in places you never had before. Your voice is changing. Your body's changing. And that's timed to be right around the time that you need to start maturing sexually. That's just life. That's puberty. So that's an example of how you had those genes before. It's not as if they just popped up into your genetic code. You've always had them, but they got turned on at a certain stage of your development. Another example is fetal hemoglobin versus adult hemoglobin. The baby, when they're in utero, the mother handles all the oxygen capture and delivery for the baby because she's doing all the breathing. So they don't need um, the hemoglobin of the baby. It needs to be able to grab as much oxygen as it can because it doesn't have lungs to get for itself. So when it transfers, there's a barrier between the mother and the baby called the placenta. When that tissue is transferring oxygen from mom and being picked up by the baby's hemoglobin needs to be much more efficient. Anyway, so it's not needed later because you have lungs and you're able to get more oxygen that way. Just another example. All right, the next few pages are practice pages. So what I want you to do is pause the recording and try to answer these. I'll read through them for you, and then I'm going to expect you to pause, and then I'll go over the answers for the first couple of slides. If mRNA has directions for hair color, what hair color will you see in the person if the brown hair is expressed and black hair is repressed? Are you going to see brown hair or black hair? The next question is, in your body, why do muscle cells and skin cells look and behave differently? Is it because they have different genes? Is it because their genes are expressed differently? Or is there no similarity in their chromosomes? Remember the chromosomes? The question is asking you, are the chromosomes not the same between skin cells and red blood cells? So take a minute, pause the video, and try to answer these two questions. Okay, now we're back. Now, in order to answer this question here, you're going to want to go back to the slide that talks about what it means if it's expressed or repressed. If we say a gene is expressed, that means the protein will be formed. So if you're talking about a color, then it will be black or brown, whichever one is expressed. If the gene is repressed, that protein color will not be displayed. So, oops, there we are, my bad. So if mRNA has the directions for hair color and the brown hair is expressed means go, means on, means yes. Okay, so in this case, the brown hair is expressed. The black hair is repressed. Re means stop, means no protein. And in the second question here, remember there's a karyotype of human chromosomes, right? There's 46 chromosomes in the human body's cells, and They have the same, every cell has the same 46 chromosomes. Okay, on those 46 chromosomes, they have the same genes, but they may or may not be expressed. So they can be turned on or off. But they're all always there. So they don't have different genes. This is false. And there is a similarity between the chromosomes. So the answer must be B. All right, the next questions. All the cells have the same genes, true or false. Here's some examples. And different genes are active in different cells, true or false. Here's some examples. So pause the video and try to answer these on your own. The answer is already here at the bottom. This is true. It says all carry the same DNA, therefore they have the same genes, because a gene is just a sequence on the DNA strand, right? It's just a sequence on the DNA strand. So if they have the same DNA, they have the same gene. And genes are active in different cells. That's true. All right, so the next slide are actual review guide questions. Remember that your review guide is extra credit. I will be going over the answers to these in class at some point, but this is the point at which you answer them here to the best of your ability. We'll go over the answers, then you'll have that part done of the review guide. There'll be review questions for every lesson. In this recap, this is practice. This is independent practice. You're going to identify the statement that's correct and fix the statements that are incorrect. So decide which of these are false and fix them. If you need a refresher of protein synthesis, this little video by Amoeba Sisters illustrates it very well. You can fill these notes out for yourself. I'm not collecting this for credit. It's just a way for you to review. And let me know if you have any questions.